Good morning, dear students. Good day, dear professionals. Happy to be here today. I'm by vocation a physical chemist working in the NMR laboratory in the same institution where Dirk Lachenmeier is from. This is a governmental food and feed control laboratory situated in Karlsruhe, close to Mannheim. We are responsible mostly for the bluish district of Baden-Württemberg, but for specialized analysis also for the rest of Baden-Württemberg. Our house is integrated. That means we run all types of analysis, and we have about 200 staff members, 50 scientists, so this is kind of medium-sized but bigger laboratory, and our house is fully accredited, so yeah, we should know what we're talking about, and we are held up to that high esteem biannually. What is more interesting, there are 36, depending on the way you count, similar laboratories all over Germany. 9 of them, the purple ones, already also have an NMR. 3 more are currently planning to buy one, to establish one. So NMR is really growing, not only in Germany, but at least in Europe. Turning to coffee analysis. As Dirk already told you, we are tasked with protecting consumers, with keeping them safe and keeping the product true. It's not reacting all the time. Yeah, there it is. So something is declared on the packaging, then it should be correct, not fraud. We can go through all this quite quickly because you are professionals and skip forward. These are aspects that are of interest, and also the ingredients if it's decaffeinated. And now to NMR. So usually we start with the roasted coffee beans or with pre-ground coffee powder because that's what the consumers mostly get. If it's not already pre-ground, then we will grind it in a high-quality laboratory mill. It's really important for analysis that is reproducible to have a clear, fine quality of coffee powder. So if you do it with a knife mill, with these Skyvy-like things, you have a wider grain distribution, and that will not be reproducible in the end. Then we extract several hundreds of milligrams of coffee powder with a solvent that can be water or that can be an organic solvent like chloroform. The suspension that we get will then be filtrated. The clear filtrate goes into a NMR tube, which is nothing more than a very precise glass tube. That goes into the, that goes into the instrument, and because you asked, one of these instruments costs about 600 to 700,000 euros nowadays, with the price rising. But you can ask Q1 in China or Jeol from Japan. They might be a tad more cheap, but the company that we are currently using, they are the market leaders and they demand that price. But as Dirk already told you, we can run about, depending on what type of analysis, we can run about 48 to 60 analysis samples per day. So one analysis takes less than half an hour in the machine. For certain types of analysis, we can do 4 samples per hour. So that is really high throughput. And we are super stable. If that machine stands in Karlsruhe or in Bogota or in Hamburg, it doesn't matter. The results taking the same coffee sample will be reproducible. But you need to start with a bigger specimen. So you need to start maybe with 200 grams of coffee, grind them, homogenize the powder, and then take your 200 milligrams that you really want to extract. In the end, you get the spectrum. That is an example of a coffee spectrum. It's a watery, an aqueous extract, pH adjusted to 6. That's more or less neutral, very slightly acidic. I won't bore you with all the chemical details, but I want you to take home that the signals are needle sharp, and that you can see hundreds of signals beside each other. So NMR is really nice to get a comprehensive analysis for complex biological mixtures in one run. Yeah? And if you go higher field strength with more powerful magnets, but which makes the price higher than 1 million euros, then you can get even more signals in close proximity. Yeah. Hop. Come on. Yeah. So first thing, as results, outcome, the question, is it Arabica, is it blended, or Canephora even pure? So this is the target substance of interest for us. That region, that methyl group, CH3 group we see here, is the one we are looking for in the spectrum. It's fast and cheap. You just make a quick extraction. Takes you maybe 10 minutes if you do a whole series, maybe 15 minutes if you just do one sample, because you need to set up everything. And then you get, yeah, Dirk has already shown you something like that. You get a spectrum with a high content or one with a very low content. So even Arabica can have a tiny amount of 16 OMC. And there are also Arabica Canephora hybrids, but they are not present in the market. So in the end, if the farmers mix up everything, that analysis might no longer be feasible. But for the time running it is. And since Canephora puts in that ingredient, you can already say if it's in, there should be Canephora in the blend. No? And we can go down reliably to 20 milligrams of 16 OMC per kilogram of coffee beans. And the accepted contamination level in industry is about 40 milligrams. So we are way below what they accept, so we can really tell them, nah, we found something that you will not accept. And the old method that was used for maybe a decade or even longer ago was HPLC, using a high temperature organic solvent extraction, and maybe you can run 3 to 8 samples per day in a normally equipped lab. We can easily run 30 to 40 samples per day, no problem. So we are faster and we produce less waste. That's the original publication, and it is my pleasure, as Dirk already told you, that we are about to finish this as an official European norm. That's the title for you if you want to look it up. And it is established and validated by 13 laboratories from 3 European countries. And it's, yes, a matter of no small pride of Dirk and mine that our house was tasked with the organization and the management of the interlaboratory collaborative ring trial. And so, yeah, we are guilty for the results. And you can expect that norm to be published maybe around the turn of the year. So, something else we can, with NMR, because the signals are so sharp and clearly separate, we can identify several more substances that might be or are definitely of interest concerning the quality and the flavor of coffee. But you need to keep in mind that with NMR, with this so-called, let me say, Volkswagen middle-class type of spectrometer. This is not a super Ferrari spectrometer. This is a reliably running. Yeah? With that type of machinery, you can go down to the maybe 10 to 20 part per million mass fraction level. So expect to be able to see and quantify analytes that are 20 milligram in 1 kilogram of roasted coffee. No lower, no flavor analysis here. Or just limited. Yeah. So now let's turn to non-targeted analysis. Yeah, we did it on roasted coffee. What I'm presenting to you right now is the work by a former doctoral candidate, now Dr. Vera Gottstein. And I'm happy she didn't find the time to do this here, so I may present her results. Maybe it's most useful for you if I explain a little bit to you what you see in here. So question was, can we identify different continental origins? And later on, can we identify or separate different countries of origin? And you see the chessboards and the balloon plots, which are called score plots. So The chessboard gives you numerical values of the true positives and false negatives. So if it is an African country with a lipophilic organic extraction, we get a bit better than 94% true positives and maybe a little less than 6% true false negatives, misaligned coffees. And vice versa, if you have a North American coffee, 96% of the samples were correctly identified by the algorithm to be North American coffees. And what you see in the score plots are many, many dots. These dots are the individual scores for the individual samples. It's a massive reduction in dimensionality. The diagram is pseudo three-dimensional, but it's a reduction for visualization. We work with a few dozens of really mathematically evaluated dimensions, or primary components. And what you can see is that the different continents, they form kind of tight clusters, and the clusters are more or less clearly separate. Even better in the watery, in the hydrophilic extract. So that's something to keep in mind. If you get a compact cluster for one real class and the other cluster is away, like two galaxies are far away, then you have a good, really, really good separation. And if they are tight, not so good. And the numerical values give you the exact numbers. What is the meaning of the balloons that you see? They are the so-called confidence ellipsoids. Depending on what level of confidence you desire, usually we set the balloon boundary to 95%. It's kind of like a multi-dimensional standard deviation. So the boundary of the balloon tells you that 95% of the samples you use to build up the model are inside and only 5% should be outside. So if you started with a good subset of authentic samples to build up the model, you can even later on expect that only 5% or fewer of new samples that you project into the model using the same mathematics will be outside. The rest should be inside. So if it's outside, it doesn't have to be wrong, but it's at least suspicious, something you want to look at quite more closely. So, PCA LDA, primary component analysis, combined with linear discriminant analysis is what the method is called. The PCA enhances the contrast between the samples with the classes, and the LDA tells you where the boundaries are. And so, we have proven so far that the differentiation from different countries of origin or continents is possible. Here again are more percentage of reliability, now in this case with external validation, so we used new samples that were not used to build up the model, completely fresh samples, and try to find out, are they aligned or classified correctly, and that are the numbers for Africa against the rest, Asia against the rest, and so on. So then next question was, can we do the same with one country against the rest of the world? And looks kind of similar or even better. So skip over that rather quickly. And we tried that out, or my colleague tried that, Brazil against the rest of the world, Ethiopia against the rest of the world, Colombia. And what is quite fascinating, and what scientists always say, that asks for more research. With Colombia, you cannot do it. So the numbers for Colombia differentiation are not making us happy. The others are acceptable to nice, but that not. Okay. So then the question was, from roasted coffee, can we differentiate organic farming versus conventional farming? I'm sorry, no. There you are. Well, so you see the balloons and the clusters of samples, they kind of intersperse. And I'm not really astonished because you lose much information due to roasting. Something else, that works out quite nicely, but I expect some of you to grin because you say, I don't need an expensive NMR. I just taste a cup of coffee for that, yeah, if it's light or dark roast. Yeah, you're sure. But you know, you have the toy, you want to play around with it. But yes, NMR naturally can differentiate if your coffee is lightly roasted or it's dark roast. But what is interesting will be the next slide. NMR can also identify between a drum roaster and a hot air roaster. So if you are a big coffee corporation using these tower-like hot air roasters, or if you are an artisanal coffee roaster like Coffee Consulate with a drum roaster, so the big guys might say, this is drum roasted, this is drum roasted. And we can say, we are not sure. We don't expect that to be drum roasted. You can see it in the evaluation. That works quite nicely. So, close to landing, summary and conclusions. What I presented to you was how we evaluate using proton NMR coffee. On one hand side, organic extraction. On the other side, hydrophilic aqueous extraction by targeted analysis, where we know what we are looking for, 16-OMC, HMF, other ingredients, and non-targeted, where we just use the fingerprinting, the profile. And yeah, NMR is quite quickly, cheaply, and easily able to quantify many analytes that are present in coffee, and NMR is also able, but not yet, definitely not yet to perfection, to classify different types of coffee, be it region, be it the variety, and so on. And to finish, I would like to, as a take-home message, to tell you a little bit about chemometrics, non-targeted analysis, because it's becoming more and more fashionable, and you should have some take-home messages. Chemometrics massively depend on the quality of your authentic model data set. You need to have a large data set. You need to have all the regions of interest. So if you have a few Brazilian coffees, maybe from the northeast, but not the rest of the country, your model will be weak. Yeah, if you do not have 5 to 6 harvest years, your model will be weak because one season was dry and hot and the other was wet and cold. So that's quite important. And you need to keep your model up to date. So the oldest samples should be phased out and new samples must be phased in because of new agricultural fashion, climate change, and so on. So you need expert knowledge to start building up a model. If your model is too small or too narrow, as I just said, it will not be robust. You put in new samples for analysis and you will get false assessments just because your model is weak. Garbage in, garbage out. Very important to keep in mind. And the final issue is the results are always just probabilities. Maybe very nice or very reliable probabilities, but there can still be the one black swan. So if something is amiss, do not accuse the guys. Ask for more information. But as Dirk already said, accusing just 1 in 1,000 is a much easier task than accusing or asking 50 from 100. So that this quickens up everything. Yeah, this is a relatively young task. It's growing, becoming more and more fashionable because computers are cheaper and so on. And the SEN, Standardization Organization, is currently working at establishing a guidance, a handbook, how to validate non-targeted methods. So if you're interested in this, keep an eye on what's going on at SEN. You will get a result in a few years. Yes, it's a big thing that we try to roll uphill. It takes time. And yeah, about the benchmark samples I already told you, but it's worthwhile because with univariate analysis, even with maybe 2 dozens or 3 dozens of coffee analytes that you can reliably quantify, it's still kind of easy to fake that. But if you go the fingerprinting way, the fraudsters would be forced to fake, a profile of hundreds or several hundreds of signals, and that's just plain uneconomic. They will not be able to do that. So if you have a good fingerprint, a reliable one, and have a good model, you really lower the risk of food fraud. And that's me at the finish. I'd like to thank Vera Gottstein foremost for her research, which was at least half of this presentation. Then we sometimes forget our lab technicians who do all the work when we do all the thinking, and they do the thinking as well. And naturally, for scientific discussion and guidance, I would like to thank our prime investigators, Thomas Kubala, who is not here, and Dirk Lachenmeier, who is here. So I'm open to your questions.
Thank you very much, Jan, for the nice presentation. And I'm opening the discussion. Questions from the audience, please.
There is one. Yeah, microphone is on the way.
Thank you. Just a quick question out of curiosity. So the principal component analysis I know a little bit about, but this distance technique for setting the limits, could you explain in 1 or 2 sentences how that works in principle? I can imagine a little bit, but...
Okay, it's called linear discriminant analysis. That means if you imagine your data were just one-dimensional, so any data point you have is on a long lane, but just on a meter stick, yeah? Then you have one group of samples here, the other here, and the linear discriminant would be the boundary that is set so that as many of the samples from one class are on the true side, on the correct side of that boundary, and for the other class the same, and that the variance inside classes is minimized. So if you go two-dimensional on a kind of a landscape, on a map, then your linear boundary would just be a straight line. If you go three-dimensional, it would be kind of like a window pane. It's always linear, so one dimension less than the ones you evaluate. Yeah? And now it gets complicated because there's much linear algebra in the background, and I don't feel really qualified to give a lecture on that. I have a nice understanding, but I think mostly that is it. You have your clusters of data, and then you just say, okay, I need either a straight line between them if I'm in a two-dimensional system, I need a kind of vertical pane or slash pane, but it's just a plane of boundary between them if they are three-dimensional. And now it's getting really complicated because, as I told you, the evaluation that we really do can be, without any problems for the computer, can be 10-dimensional or 20-dimensional or whatever. It's an orthonormal vector space, so all of the principal components are oriented orthogonally toward each other. But I think most of you, I at least, am unable to imagine visually something that is even 4-dimensional. Yeah? But in linear algebra you can do it, and if you have enough time you can solve it just with a block of paper and a pencil. The mathematics is not very high, but it's a massive amount of data you have to process. Yeah? But that was so many buzzwords and so little explanation, I fear.
But the main criterion is the internal variance of each group. Did I get that correctly, basically?
Yeah, that's it.
Okay.
So
cool.
If you, that's quite a good follow-up question, because if you have a situation where you have two classes, but the variance inside at least one of these classes is still quite big, and the classes, they intersperse, then PCA, LDA can even model up issues and not clarify separation. So what you want and what you're hoping for, what we are hoping for in the lab, is kind of tight clusters with small inside class variance, but a bigger between classes variance. Then you're really happy, because then you can say there is the boundary, there's lots of empty ocean, data ocean, so to say, in between. then you can easily say, if your sample is halfway to the border, then it's really not typical for the class it was declared to be. Yeah? But even so, still a probability, not an absolute truth.
Thank you.
Okay, thanks for that thorough explanation. Any other questions? Maybe one from my side. You mentioned you were able to differentiate different cultivars or different species depending on the availability.
Yeah.
I assume these were pure samples, right? So you differentiated pure samples.
We did also a type of, was it 7 or 9 samples in the Sen ring trial? We systematically blended them.
Okay, and that would be the question. What is your cutoff? Your rough estimation of the cutoff, where you could say this is blended?
So. As I said during the talk, the so far accepted level of 16 OMC in Arabica or in coffee sold as pure Arabica is 40 milligram per kilogram of coffee beans, roasted coffee beans. The issue being that it was always claimed, yeah, there could be some cross-contamination from some leftover beans in the drum, whatever. Basically, if you have a roaster with a tidy roasting workshop, the cutoff should be lower because the absolute majority of Arabicas has a 16 OMC content that is way below 40 milligrams per kilogram. It's usually even below what we can easily and quickly quantify with our 400 megahertz machinery. We can always invest more measurement time and then lower the threshold down to which we get precise results, but then it becomes expensive because measurement time is money. Yeah? So, and something that was asked earlier this morning, can we say something about how much of a percentage of Canephora is blended in? Yes and no. Yes, we can, if we have reliable samples of the original coffees that are blended in. No, we cannot if we just have this package of commercial off-the-shelf coffee and we find maybe 300 milligrams of 16 OMC in that package. That is definitely not a pure Arabica, but without knowing the 16 OMC content of the Canephora that was blended in, it can be 30%, it can be 60%, it's just not easy to say because the variance of 16 OMC content in Canephora is from a few hundred milligrams, say 600, 800 milligrams per kilogram, to way beyond 1,000 milligrams. So without knowing your Canephora starting, can't say.
But it would still be worth in terms of a suspect screening. So you would know there's something suspicious with a sample, and that would be an additional analysis if necessary.
Yeah.
Okay.
Definitely.
There's
You
another
could
question.
ask for the authentic sample. Maybe you get one.
Thank you very much for your very interesting lecture. Did you say, if I am not wrong, that you cannot make a differentiation between the organic and the no-organic one? I think that for the processing, you can just make the differentiation with the chemical marker that you are using, and also you can do it for the species and so on. But if I am not wrong, I think that you mentioned that you cannot make a differentiation between the organic and non-organic one. It may be because the chemical marker that you have selected, or do you believe that you need just to move anyway to another technology just to make the differentiation, or we are not able just to make this differentiation?
Okay, so your main question seems to be, can we differentiate organic versus conventional farming? As said, no, we cannot. So why can't we? I think, start differently. When we do chemometrics, we do kind of comprehensive fingerprinting. We do not in the beginning at least, we do not ask for individual markers. We just have two classes of which we are confident, highly confident that they are authentic organic or authentic conventional farming, and then we tell the machine, hey, look if you can find any difference anywhere. And as we already heard, coffee contains thousands of ingredients that are quantifiable, maybe hundreds of them that are reliably quantifiable for NMR because the sensitivity does not go way down like chromatography. And I think it might be possible to differentiate when you use green coffee. But when you use roasted coffee, you lose some information. And the issue is, what is the difference from organic to conventional farming? It's agrochemicals and artificial fertilizer. I'm not too sure if they will have such a high impact on the metabolome, and what we see is the metabolome. And if you then roast that metabolome, then you lose some information. So I'm not sure if it's economically efficient to try to differentiate between organic conventional farming using NMR analysis. It could be interesting when you have green coffee, but the problem is for food control, what we do, what is our day-to-day work, food control focuses on roasted coffee. So for big coffee traders, if you are a coffee trader and you don't want to get frauded upon by getting conventional coffee for organic prices, so to say, then you might be interested in checking, hey, give me dozens, hundreds of samples green beans, and then we look at it again. Yeah. But that's not our job, so alas.
That's right. Yeah.
Does it help you in the sense of an answer?
Yes, I agree.
Okay.
Thank you.
You're welcome.
Okay, are there any other questions? So if that is not the case, then please join me in thanking Jan for his great presentation. Thank you very much. And I still have something to hand over.